Okay, great. So I want to go back and remind us that before we talk about the Delta variant, that we are really in this very fortunate position that we do actually have 9 active vaccine candidates for COVID-19. There are 3 that are in this country. Moderna is authorized. Pfizer is approved now as of yesterday. Johnson & Johnson is authorized. And then there are 6 more from around the world. And these are all the references where you can find all the data for the phase 3 clinical trials for all of those. Now, remember, 6 of them involve the spike protein of the virus. So the spike protein is the little piece that sticks out from the SARS-CoV-2 virus, connects with the host cell, and 6 of those vaccine candidates either involve genetic material that codes for the spike protein in 5 out of 6, and one of them is actually giving the spike protein with an adjuvant. That's the Novavax vaccine. 3 of the vaccines that are available, 2 in China and 1 in India, are inactivated whole virions. And in terms of those vaccines that involve the spike protein, which are more relevant to the United States, there are 2 that are mRNA vaccines. All that means is that we give the mRNA in a lipid bilayer, and the mRNA comes into the body, and then you translate it into protein, the spike protein, and then you raise an immune response against that spike protein. And then 3, and that's Moderna and Pfizer, of course, and then 3 of them, Sputnik 5, AstraZeneca, and Johnson & Johnson, involve the adenovirus DNA. So it's the DNA genetic material surrounded by an adenovirus as the vector. The DNA comes in, gets transcribed to mRNA, and then the same process, translated to protein, and you raise an immune response. And then the Novavax, which is not yet authorized here, is just giving the protein itself with a adjuvant. Now, we really, to understand the Delta, have to remember the immune system. We have to remember what cellular immunity is. And I think that will help us realize that we will get through this pandemic with these vaccines. So there are 2 types of cells that get stimulated by the vaccines. There are B cells, which are your usual cells that make antibodies. They actually go into the bank and form memory B cells. And it's very natural and normal for antibodies to wane in the body. That's what's happening right now in our world, since we've had the vaccines a little bit of time ago. But that memory B cells are there to make more antibodies if they ever see the virus again. And we're going to talk about that. T cells are also generated by the vaccines, and they're divided into T helper cells and cytotoxic T cells. And importantly, T cells are really what protect us against severe disease. And how do we know that? Many papers on this, including a lot here from UCSF, from the Gladstone Institute at UCSF, but really that T cells, we knew this from other infections, but many papers show us that it's really our T cell immunity that protect us from getting severe disease with COVID-19. So when we hear some of these familiar refrains that we're going to get, that we're still being protected against severe disease, but maybe less effectiveness against mild disease, that really is thanks to our T cells. And we'll talk about that more. Remember, all the phase 3 clinical trials of all the vaccines, including the 6 spike protein candidates, took the trouble, as shown in column 4 here, to measure T cell responses. We do get strong CD4 and CD8 responses to these vaccines in the phase 1-2 clinical trials of these vaccine candidates. Thus, on the column 6, you can see that there was a lot of protection from severe disease in the clinical trials. In fact, it was almost 100% across all the trials, hospitalizations and COVID death. That is really a function of our T cell immunity, and that's what's being preserved now, even in light of the Delta variant. There was and remains variability across the variants for more mild disease, and that actually has to do with antibody responses, which is your first line of defense in the nasal cavity. This is true of the inactivated whole virions as well. All 3 candidates, the Covaxin product by Bharat Pharmaceuticals in India and the Sinovac and the Sinopharm, they have less responses against mild disease, but they have still strong protection against hospitalizations with COVID-19. That is really a result of T cell immunity. So, sorry, this got out of sync. So one thing that we should remember about the vaccines, and I'll go back to children, is that they were all authorized in December and early January, December 2020 and early January. The ones that were authorized in December were the Moderna and Pfizer vaccines. And then yesterday was quite a significant event, which is that the FDA took the emergency use authorization and moved it to a full approval of the first COVID-19 vaccine, which is the Pfizer-BioNTech product. And this is really important to remember. Oh, unfortunately, it's called Comirnaty, which is kind of difficult to pronounce, but important to remember that what they did yesterday was approve it fully for 16 and older, but the vaccine is still continuing under an emergency use authorization for children between 12 and 15 years of age, and also approved a third dose for immunocompromised individuals. Now, this is the data of the Pfizer vaccine in children. This was published in the New England Journal. And remember, it was done in quite a small group of children, just 2,260 adolescents. 1,130 got the placebo, 1,000, essentially half and half got placebo in the vaccine. And there were only 16 cases of COVID-19 in the 12 and 15-year-old group. And all of those were in the placebo group. Antibody responses were stronger than in adults. This is still under EUA because it was a EUA authorization was issued on May 10th, and it was not moved to approval yet on August 23rd because the FDA wants 6 months of data on these participants. And we only have we have less than that now. So longer term data is needed on the 12 to 15-year-olds before it's fully approved. The reason that there is concern is that essentially there have been cases of myocarditis seen mostly in young males, mostly after the second dose. These myocarditis cases, as described on July 9th by the CDC, are mild in general, and though a couple of children did need more intensive care. And because of that, because of the myocarditis issue that was also seen with the Moderna vaccine in their 12 to 17-year-old trial, these children need to be followed longer to confirm safety before full approval. And the other thing that the FDA did was ask Pfizer and Moderna both to expand their numbers of the children that are being studied in the 5 to 11 trials away from that very small number of 2,260 that they were doing for the 12 and 15-year-olds, and they were planning for the 5 to 11-year-olds. They have asked both companies to increase the 5 to 11-year-old children group study to 5,000 to be able to detect more rare side effects. And so both companies agreed. And also the 12 to 17-year-old trial for Moderna has been expanded to 5,000 on the CEO's request. And what the Pfizer CEO said yesterday when their vaccine was approved is that they think that the 5 to 11-year-old data will be completed in September. So quite coming up for them. They said that it will take longer for the 6-month to 5-year-old data. So, okay, so then the next question that comes up is, do the vaccines work against the variants? And really, we have to understand T-cell immunity to answer that question. The reason that it's important to understand immunology before dismissing the vaccines work against variants, which some have done, I think, who don't have maybe a more fundamental understanding of immunology, is that T-cell immunity is very robust. It's very in breadth. And to put it very simply, the T-cell immunity is preserved against all the variants. So let's remember our variants. They used to be called by names, then they were called by numbers, and now they're called by the World Health Organization names, the alpha, beta, gamma, delta. Delta was what had arisen in India in early March and really caused a terrible, terrible second wave. That was in the face of 4% vaccination rate in India at the time. And this Delta variant has not only arrived in our shores, but is absolutely the dominant variant. Over 90% of our strains are Delta. It's very transmissible. It causes high viral loads. And because of that, it has a very short incubation period. So it really is transmissible. So what are we seeing with Delta in terms of T-cell immunity? this is the simplest way that I can put it. Essentially, Delta has 13 mutations across the spike protein, and the spike protein has 100 different epitopes that T cells work against. What that means is that there's 100 different T cells essentially that line up across the spike protein. So if you have 13 mutations across the spike protein, you have lost some of the T cell epitopes to which they respond, but you have lost only a minority. There are still multiple T cells that line up against that spike protein to work against the variants. And this has been shown in several studies, both from the La Jolla Institute of Immunology in San Diego, from UCSF Gladstone Institute, that T cell reactivity is intact against all the variants, including Delta. And it really has to do with the fact that there's such an in-breadth T cell response across the spike protein. This is not only shown in vitro, there's further studies by NIAID, and the AstraZeneca trial with the gamma variant, for example, showed that 13 of the T cells were knocked out, but 75 were still intact across the spike protein. But this has been shown in vivo. And what I mean by that is that the public health data from England, and we're going to go over this a little bit more, against the Delta variant, still shows that there's high effectiveness in the UK, in Canada, in the US, and in Israel against hospitalizations and severe disease. That really speaks to our preserved T cell immunity, even in the face of the Delta variant. So to ask this question, or to answer this question, are we seeing waning vaccine effectiveness? We really do have to divide our outcomes into the two outcomes that the two types of immunity affect. We have to divide our outcomes into severe disease, where T cell immunity is the main effector to reduce severe disease, and then into B cell immunity, which produces antibodies. So to explain that a little bit more, vaccine effectiveness is simply not just a response that has to do with the host and the virus and the vaccine. There are multiple factors that go into vaccine effectiveness. One is that if you have high levels of circulating virus, then a vaccine, you're going to get more breakthroughs, whether you have a highly effective vaccine or not, because you just are seeing a lot of virus. If you are in a household with someone who has COVID, you can have a very good vaccine on board, but you're getting a lot of viral inoculum. So there's demographic factors, there's vaccine access factors, and the type of vaccine, and also importantly, the timing between doses, which we'll talk about in a minute. There are immune factors, there are viral variant factors, which is only one aspect of vaccine effectiveness. And then there's host factors, including age and previous infection, which will give you immunity, immunocompromise, and underlying health conditions. So there's many factors that go into vaccine effectiveness. And when we look at vaccine effectiveness in light of the Delta variant across multiple countries, England, Israel, Qatar, US, and the UK and Canada, which isn't on this slide, efficacy against hospitalization is remaining highly intact. So it is essentially in the 90s. We'll talk about the US study in older patients that went down lower, about 78%. Otherwise, hospitalization, efficacy against severe disease and hospitalization is remaining high. Why? Mostly above 90%, because of T cell immunity. In terms of symptomatic disease or getting infections, getting mild symptomatic breakthroughs, even that, even the effectiveness against those are essentially the same in the UK and Canada, which are around 80%. And in the US, they're around 78%. But there is an outlier study from Israel that shows the effectiveness against mild infections or symptomatic disease with a mild breakthrough vaccinated is 40%. The important thing about that Israeli study is we don't have the methods, it's not peer-reviewed, and it's very difficult to try to understand their methodology without more information from Israel. The UK and Canada studies are full publications where we have these high levels of effectiveness, even against symptomatic breakthroughs. But there's another reason why Israel and the US could be differing from the UK and Canada. Why could there be more symptomatic breakthroughs with Delta in the US and Israel as compared to the UK and Canada? I actually think it has to do with the duration between doses. So there are two reasons why we could be seeing more symptomatic breakthroughs with Delta. One is could be the higher viral load. But the second is that, again, antibodies naturally wane with time. There are healthcare workers, nursing home residents who got their vaccine in December. It is now August. And there are others who got their vaccines in March and April. It is natural for antibodies to wane with time. That's not a glitch of the immune system. That is what it does. You can't keep all your antibodies in your bloodstream for every infection and vaccine you've seen. Otherwise, your bloodstream would be as thick as paste. So this is going to happen. But what actually leads to an increased antibody titer? A Nature paper shows us that it's increasing the duration between doses, which is actually a general principle of vaccinology. We've never actually given any vaccines, the 2 shots 3 weeks apart. And so the UK and Canada both decided on a first dose first strategy, first giving the first dose and then going out to 12 weeks. They reduced that to every 8 weeks with Delta. But because they had a longer interval, they likely have higher antibody production against they have higher antibody production because of this longer duration between doses, which is not a strategy. We chose to follow the clinical trial 3 weeks with Pfizer in the US, and also Israel did the same thing. There was a study from the Mayo Clinic system that showed that there's less mild symptomatic breakthroughs with the Moderna vaccine than with the Pfizer vaccine. And the study authors postulate that's because Moderna is given at a 4-week interval, not a 3-week interval. Even having 4 weeks is better than having 3 weeks in terms of boosting your immune response with antibodies. So it is normal for antibodies to wane. That could be why we're getting more symptomatic breakthroughs with Delta, but luckily this is our T cells responses are staying intact. What can we do about waning antibodies? Well, importantly, and this is an important concept, the best, and it's hard to hear, but we absolutely have a way to make more antibodies because that's what memory B cells do. Our memory B cells are produced by the vaccines if we're immunocompetent. We have lymph node data and we have bone marrow biopsy data that shows that we form strong memory B cell islands with the vaccines. And what happens when you first see a virus is that your antibodies go up and then they naturally wane. Again, totally normal. And then the next time the immune system sees the virus, you actually produce even higher levels of antibodies. Now, importantly, 2 papers, one from OHSU and one from the University of Pennsylvania, show us that memory B cells don't produce antibodies against the ancestral strain. If they see the Delta variant, they will produce antibodies that are directed against the variant at hand. Because again, the Delta variant is not that different from the ancestral strains, about 13 mutations across the spike protein. And the memory B cells form a blueprint, but they don't have inherent antibodies in them. They will come out and form antibodies against the variant at hand. So memory B cells will adapt their antibodies to the variant that they see. And that is why giving boosters for immunocompetent patients may not be the best idea, because what does the booster or a 3rd shot do in immunocompetent patients? Remember, the current booster that we have is the Pfizer vaccine or the Moderna vaccine with the original mRNA coding for the ancestral strain. So if you give that booster, a 3rd shot to immunocompetent individuals, what you're going to do is develop antibodies against the ancestral strain. If you see Delta in your nose and you're fully vaccinated, you will develop antibodies against the variant at hand. So this is the complexity and the in-breath response and the amazingness of our own immune system. And I'm not sure that boosters to the immunocompetent is going to help. Do I think that we need boosters for immunocompromised? Yes, I do. And that is because they're not able to form the memory B cells that you need. You need really another booster shot to form what's so important with your memory B cells and T cells. So currently the CDC is recommending that moderately to severely immunocompromised patients do receive a third booster shot. That is again on the approval package insert for the Pfizer vaccine. And this includes people receiving cancer treatment, organ transplants, and are still on immunosuppressive, stem cell transplants, primary immunodeficiencies, advanced or untreated HIV infection, or are on immunosuppressive drugs. And this is how we're defining, at least in our HIV clinic that I am the medical director for, called Ward 86, that anyone who has a CD4 cell count less than 200, virologically non-suppressed, is who we're giving booster shots to. And we're also giving that if they've had a history of AIDS, sort of using provider judgment. But again, giving boosters to the immunocompetent instead of focusing on our unvaccinated individuals in this country, and importantly, unvaccinated people worldwide. So right now, of 5 billion vaccine doses given out, only 2% of those vaccine doses have been given out in low-income countries. You are 50 times as likely to have been given a vaccine if you live in a high-income country than a low-income country. This degree of incredible global vaccine inequity this long into our vaccines being authorized, I think is a moral and ethical blight on our country, actually, and on all rich countries. And the only way to stop variants and to get through this pandemic is global vaccine equity. Okay, do vaccines reduce transmission? Well, in a way, it is less so with Delta. And I think there is a pre and post Delta about vaccines reducing transmission. So we always knew that it was biologically plausible, not just plausible, but vaccines do get in, antibodies do get into the nose. Your IgA immunoglobulins are stimulated by vaccines. IgG is stimulated by vaccines, and IgG actually are found in high levels in the nasal mucosa. And as those antibodies wane, the IgA and IgG, you're more likely to get infected in your nose. So what will that lead to? Mild symptomatic breakthroughs, but it will also lead to more transmission. And I think that is what's happening with the Delta variant. These were all the studies that showed us before the Delta variant how much asymptomatic infection was reduced. In fact, study after study after study showed that not only do the vaccines reduce symptomatic infection, but massively reduce your ability to even have it in your nose, harbor it in your nose at all, which is why that masks were taken off for the vaccinated on May 13th by the CDC. Then what happened was a Provincetown outbreak, and the CDC on July 27th put back masks for the vaccinated, maybe not for the right reasons, because they cited this Provincetown outbreak study and said that vaccinated people were as likely to transmit as unvaccinated people. That actually was not shown by the study, as I explained here. So I think what ended up getting lost in that Provincetown outbreak study, where it was a kind of menosis with men weekend, mostly a lot of people coming in with mixed vaccination status, even though Provincetown itself had a high level of vaccination. So a lot of mixed people. It happened to be raining, a lot of inside activity, windows were closed, no masks, and importantly, lots of exposure, lots of intimacy, lots of things happening. And there were high levels of symptomatic breakthroughs, but they really were mild. And I think it was a real stress test for the vaccines that severe disease was very low. Only 3 people were hospitalized and they got out of the hospital very quickly. So it was really a great stress test for how well the vaccines work against severe disease. However, what happened was that at a single point in time, the cycle threshold of the PCR tests in vaccinated people versus unvaccinated were the same. Now, the problem with using that as a marker for infectiousness is that that was just a single point in time. So what is a cycle threshold? The cycle threshold is the number of times that the PCR machine has to go through before it triggers the machine to say that there's a positive test. And so the higher the cycle threshold, the lower the viral load. And just because the cycle threshold was the same at one point in time, the Singapore study showed us the next day, these were symptomatic breakthroughs with Delta, that the cycle threshold value was the same at one point in time. But if you did serial viral load testing, the viral load went down much more quickly, as shown on the right, in those who are vaccinated, which of course makes sense because your antibodies and even T cells are in the nose will come in and hobble that virus. And then importantly, what about culture data? Because to say that something is infectious by a PCR test is not accurate unless you can actually culture the virus. And this was just a study from yesterday, actually, that shows us that with Delta breakthroughs in vaccinated healthcare workers in the Netherlands, there were 161 breakthroughs, that when they cultured the virus, the virus was less likely to be culturable or infectious in the healthcare workers that had Delta breakthroughs compared to, of course, unvaccinated people with natural infection. So the bottom line is that though vaccinated people are probably spread with symptomatic breakthroughs, which is a difference with the Delta virus, it's definitely not as much as unvaccinated infections. The CDC probably jumped the gun before they incorporated culture data and also serial viral load data from these other studies. This is the question about asymptomatic vaccinated people transmitting Delta. And this is the closest we can get to good case control studies. This is a Singapore contact tracing study. And if you go to this website, which I'll put on the slides when I send them out, vaccinated asymptomatic people were very unlikely to contact trace to spreading it. So this is good news for people who are who are vaccinated and asymptomatic in terms of being still pretty safe from spreading it, which is why the CDC is still not recommending asymptomatic testing. People who are asymptomatic after vaccination, unless you've had an exposure. They did change that with Delta, but just mass testing asymptomatic people after vaccination is still not recommended. Symptomatic breakthroughs are being monitored by states, and they're also being monitored in cohort studies by the CDC. And then the CDC is also monitoring severe breakthrough infections with the vaccines. The good thing about what's going on with severe breakthrough infections is, despite the Delta variant rising, these numbers of the number of Americans that are experiencing severe breakthrough infections are still remaining very low. So this is just data from this morning. This is updated quite regularly from their breakthrough infection website. But out of over 168 million Americans who are fully vaccinated against COVID-19, there have been 6,059 hospitalized breakthroughs, and that represents a 0.003% chance of having a severe breakthrough infection. And then in terms of deaths from COVID, it's a 6 in 1 million chance if you've been fully vaccinated, so very low rate, and that isn't actually changing with Delta. I think speaking again to our T-cell immunity to the vaccines. So a couple of other things I want to say about the immune response. When I said that memory B cells are triggered by the vaccines, and we know that from scientists have biopsied lymph nodes and seen memory B cells formed with the vaccines. They've biopsied bone marrow with natural infection and found the formation of memory B cells. And then also we can find memory B cells in the bloodstream. Well, how long do memory B cells last? I do want to tell you about this really nice study, which is from patients who had memory B cell immunity lasting after 90 years to the influenza pandemic. This is this top study, this top line here, but they took people who were between like 3 to 10 years old in 1918. So they had been born in 1915 or 1908. And then this is a 2008 Nature paper. They actually found individuals who had survived, survived the influenza pandemic, found these 90 to 101-year-olds, and checked that they still had memory B cells in their bloodstream, which they all did, to the original influenza strain, took their memory B cells, put it in a test tube with the influenza strain, and found that those memory B cells could form strong neutralizing antibodies against the original influenza strain from 1918, and those neutralizing antibodies protected mice from infection. So really, memory B cells last a long time. We already know that memory T cells last a long time. There's multiple papers that show that measles vaccination, for example, as children, you have durable T cell immunity for 34 years and counting. And then we know from the SARS original pandemic from 2002, 2003, that people who develop T cell immunity immunity, people develop strong T cell immunity, and that 2020 Nature paper showed us that they still have strong T cell immunity 17 years later. So if we can just get transmission down, which again is more about global vaccine equity as well, then coronaviruses themselves don't actually mutate that quickly. They just mutate a lot because we have a lot of transmission. But coronaviruses have a more fastidious polymerase, much more fastidious than the influenza polymerase, much more than the HIV polymerase. They have a strong proofreading mechanism, and if we can get transmission down, we will not have continual evolution of variants, which is why cannot stress enough global vaccine equity. So there's a simple premise why our vaccines will work against the variants. It's very simple. Memory B cells, if they see the virus again, and if they see a variant, they will produce antibodies directed against that variant. Memory T cells produce multiple in-breath responses across the entire spike protein. So knocking out some will not knock out your T cell response. So it really is a matter not of, I think, giving boosters to fully vaccinated immunocompetent individuals. It's not a matter of developing variant-specific vaccines. I think the entire problem that's going on right now is we did not have enough vaccinated adults in the United States when the Delta variant hit. And counties and countries with high rates of vaccination did much better with the Delta variant, including, for example, San Francisco County, which already our cases are coming down quite quickly after the Delta variant hit because we had higher levels of immunity. The same thing happened in the UK versus Israel, where there's more vaccine hesitancy among certain groups in Israel. So it really is about getting the unvaccinated vaccinated. And I think that's where we should be concentrating our efforts. And I will end here and see what questions you have.